The determination of the order of nucleotides or base sequences in a DNA or RNA molecule is called sequencing. Short DNA sequences that has a single occurrence in the human genome and whose location and base sequences are known. Such places of genome are called. Such places of genome are called sequence tagged sites. Sequence tagged sites derived from complementary DNA, cDNA, is called as expressed sequential tags. Expressed sequential tags. Sequences which are detectable by polymerase chain reactions and are useful for localizing and orienting the mapping and sequence data reported from many different laboratories and serve as a landmark on the developing physical map of the human genome are called as sequence tagged sites. Sequences of varying lengths that occur in multiple copies in the genome and it represents much of the human genome are called as repetitive sequences. Are called as repetitive sequences. Map of locations identifiable landmarks and DNA restriction enzyme sites or genes regardless of inheritance is called physical maps. Stretches of DNA that do not code for genes, also called non-coding DNA, which may have regulatory and other functions, such DNA is called junk DNA. Common bacterium that has been studied intensively by geneticists because of its small genome size and normal lack of pathogenicity and ease of growth in the laboratory is Escherichia coli. Advantages of HGP in the areas of healthcare. Identification, mapping of genes responsible for genetic diseases that helps in diagnosis, treatment and prevention of these diseases. Advantages of HGB with reference to disease and prevention. Earlier detection of genetic predispositions, genetic predispositions to disease, rational drug design, rational drug design and gene therapy. Earlier detection of genetic predispositions to disease, rational drug design and gene therapy. A new area in medicine which is characterized by looking into the most fundamental causes of disease, then treating the symptoms by using advantages of HGP, that is molecular medicine. That is molecular medicine. 99% of the base sequence among human is the same. Assuming human genome has 3 into 10 to the power of 9 base pair, and how many base sequences would there be? Differences 3 million base pairs. Percentage of differences of DNA which make very individual unique in their phenotypic appearance 0.1%. One aims to find out the genetic differences between two individuals or among individuals of a population. Sequencing the DNA every time would be a daunting and expensive task. True or false? True. DNA fingerprinting is a very quick way to compare the DNA sequences of any two individuals. True or false? True. Microsatellite code for some special protein. True or false? False. Many satellite code for protein. False. They do not code for any protein. DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called as called as repetitive DNA. Called as repetitive DNA. Are separated from bulk genomic DNA as different peaks during density gradient centrifugation. Oh my God, wait. DNA fingerprinting involves identifying differences in some specific region in a DNA sequence called as repetitive DNA sequences, no doubt, repetitive DNA sequences, because in these sequences, a small stretch of DNA is repeated for many times, repeated for many times. Here it is in DNA fingerprinting, repetitive DNA, repetitive DNA are separated from bulk genomic DNA as different peaks during density gradient centrifugation. If a DNA fingerprinting during density gradient centrifugation, the dash DNA forms a major peak. The bulk DNA form a major peak and other small peaks are referred to as satellite DNA. Satellite DNA. Depending on the base composition, AT rich or GC rich, length of segment and number of repetitive unit, the satellite DNA is classified into many categories such as microsatellites, mini satellites, microsatellites and many satellites. Likewise, micro and mini satellite do not code for any protein. Yes, they do not code for any protein. Microsatellite, mini satellite form a large portion of human genome. Yes, they form a large portion of human genome. Microsatellite, mini satellite show high degree of polymorphism and form the basis of DNA fingerprinting. True or false? True. DNA from every tissue such as blood, hair, follicle, skin, bone, saliva, sperm form an individual show the same degree of polymorphism, which they become very useful identification tool in forensic applications. True or false? True. Polymorphisms are inheritable from parents to children. Polymorphs can be inherited from parents to children. True. Polymorphisms are inheritable from parents to children. DNA fingerprinting is the basis of paternity test in case of disputes. These sequences normally do not code for any proteins, but they form a large portion of human genome. Is that? That's what we are talking about. That is microsatellites and mini satellites. Microsatellites and mini satellites. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Microsatellites and mini satellites. These sequences, microsatellites and mini satellites are, since DNA from every tissue, such as blood, hair follicle, skin, bone, saliva, sperm from an individual show same degree of polymorphism. They show the same degree of polymorphism. They become very useful identification tool in forensic application. Basis of genetic mapping of human genome as well as DNA fingerprinting is polymorphism. Variations at genetic level are called as polymorphism. We call polymorphs. Polymorphism arises due to mutations. Cells that generate gametes in sexually reproducing organisms are called germ cells. New mutations may arise in individual in somatic cells or germ cells. So might be both somatic or germ cell, but only germ cells can be inherited. Somatic and germ cells. If a germ cell mutation does not seriously impair individual's ability to have offspring who can transmit the mutation, it can spread to the other members of population through sexual reproduction. Allelic sequence variations has traditionally been described as a DNA polymorphism. If more than one variant allele at a locus occur in human population with a frequency of greater than 0.01, inheritable mutation is observed in a population at high frequency. It is referred as DNA polymorphism. The probability of such variation to be observed in dash non-coding DNA sequence would be higher, as mutations in these sequences may not have any immediate effect or impact on an individual's reproductive ability. These mutations keep on accumulating generation after generation and form one of the basis of variability of polymorphism, that is mutations on non-coding DNA. There is a variety of different types of dash ranging polymorphism. There is a variety of different types of polymorphisms ranging from single nucleotide change to very large scale changes for evolution and speciation. Such polymorphisms play a very important role. DNA fingerprinting was initially developed by scientist Alec Jeffrey. In DNA fingerprinting, Jeffrey used this DNA as a probe, satellite DNA. He used a satellite DNA. Jeffrey used satellite DNA as a probe. Probe used in DNA fingerprinting, which shows very high degree of polymorphism, that is satellite DNA. It was called variable number of tandem repeats, that is satellite DNA. VNTRs expand variable number tandem repeats. The fingerprinting technique as used earlier involved in southern blot hybridization technique. Here, southern blot hybridization technique using radio labeled VNTR as a probe, and it included separation of DNA is called isolation of DNA. Enzyme used in the digestion of DNA. Enzyme used in the digestion of DNA, we use restriction endonucleases. Restriction endonucleases. We use technique used for the separation of DNA fragments, that is gel electrophoresis. Transferring of separated DNA fragment to synthetic membranes such as nitrocellulose or nylon is called as blotting. Hybridization of DNA by using labeled what? VNTR probes. Hybridization of DNA using label radioactive VNTR probes. We can say here radioactive probes which are complementary to that VNTR. Detection of hybridized DNA fragments by itself autoradiography. Isolation of DNA, digestion of DNA by restriction endonuclease, and transferring of separated DNA fragments to synthetic membranes such as nitrocellulose or nylon, detection of hybridized DNA fragments by autoradiography, hybridization using labeled VNTR probes, separation of DNA fragments by gel electrophoresis. Put them in order. Let's start. First of all, we'll come from here, isolation of DNA. We should start from here, isolation of DNA. Then digestion of DNA by restriction endonucleases. That is the second one. Digestion of DNA by restriction endonucleases. So next, separation of DNA fragments by gel electrophoresis. Separation of DNA fragments by gel electrophoresis. What next? transferring separated DNA fragments to synthetic membranes such as nitrocellulose or nylon. Number 3. What next? What next? Hybridization using labeled VNTR probes. Excuse me. Then finally, detection of hybridized fragments, DNA fragments by autoradiography. This is the biggest question as this is very, very, very important. This is a sequence finally. Simply we say the sequence is 1, then 2, then 6, 3, 5, then 4. The one and only area as a question in Neet from this particular area, other than express sequential tag and sequence annotation. The VNTR belongs to a class of satellite DNA referred to as mini satellite. If in VNTR a small DNA sequence is arranged randomly in dash randomly in many copy numbers. The copy number varies from chromosome to chromosome in an individual. The number of repeat VNTR show very high degree of polymorphism. As a result, in size of VNTR varies in size from 0.1 to 20 kb. After hybridization with VNTR probe, autoradiogram gives many bonds of differing sizes. Why? Why? Because VNTR of individuals are not same. Bands of autoradiogram of DNA fingerprinting give a characteristic pattern for an individual DNA. It differs from individual to individual. Why? Why? VNTR of individuals are not same. Or we can say VNTR show high degree of polymorphism. VNTR show high degree of polymorphism. VNTR hybridization bands and autoradiogram similar in at least in monozygotic twins. We call them as identical twins. The sensitivity of the DNA fingerprinting technique has been increased by use of polymerase chain reaction. DNA from a single cell is enough to perform DNA fingerprinting analysis because of this technique. polymerase chain reaction, which we can take multiple copies of sample DNA, not to lose the sample. DNA fingerprinting used in that we can use this in forensic science for crime aspects, then population diversity, then genetic diversity. Currently, many different proofs are used to generate DNA fingerprints. True or false? Yes, that's true. That's it. Thank you.